Hello and welcome to my science tutorials. In today's video, we want to look at the introduction to biology multiple choice questions, where we'll be looking at the WAEC biology past questions that has come on introduction to biology. So without further ado, let's begin. So let's look at the objective questions that came on introduction to biology. Question number 1. Temporary slides prepared to be examined under the microscope are always covered with a cover slip in order to A. Avoid breaking the slide. B. Make the object appear clearly. C. Prevent the object from falling. And D. Avoid wetting the objective lens. So what is the answer for this question? So the answer is D. Avoid wetting the objective lens. So when we prepare temporal slides, we used to cover them, or we have to cover them with cover slip in order to avoid wetting the objective lens. Question number 2. A prepared slide to be observed under the microscope must be on the, this is an obvious question on a microscope. So when you prepare a slide, you need to put it onto the stage or mount it onto the stage. Use the clip holder to hold it onto the stage firmly before you can be able to observe using the objective lens. Question number 3. Which of the following parts of a light microscope contains a lens? A. The diaphragm. B. The eyepiece. C. The fine adjustment. D. The nose piece. Now the question is asking which of the following parts of a light microscope contains a lens. The only part of a microscope that contains a lens from these options we have over here is B, the eyepiece. So the eyepiece of a microscope contains lens. Question number 4 is just a repetition of the first question. The main aim of covering a prepared slide with slip before viewing an object under the microscope is to A. Improve upon the image form. B. Prevent crashing the section of the slide. C. Prevent the section from drying up. And D. Avoid wetting the objective lens. So as we saw in the first example, the main reason why we cover a prepared slide with slip is to avoid wetting the objective lens. So the answer over here is D. Question number 5. The internal structure of a chloroplast was revealed by A. Electron micrograph. B. The light micrograph. C. Photographs. D. X-rays. Now, the internal structure of a chloroplast was revealed by the use of the electron micrograph. Question number 6. The scientist who invented the microscope was A. Carolus Linnaeus. B. Antony van Leeuwenhoek. C. Isaac Newton. And D. Charles Darwin. Now, the answer for this question, the scientist who invented microscope, is Antony van Leeuwenhoek. So the answer for question number 6 is B. Question number 7. A temporal slice prepared to be examined under a microscope are always covered with a cover slip in order to, so this is just a repetition of the first and then the fourth question, but this is also a question that came for WASSCE 2013, so they repeated quite a couple of questions. So to take question 7 again, the temporary slice prepared to be examined under a microscope are always covered with a cover slips in order to A. Make the object appear clearly. B. Avoid wetting the objective lens. C. Prevent the object from falling. And D. Avoid breaking the slide. So the answer, as we already know, is B. Avoid wetting the objective lens. Question number 8. Which of the following statements about a control experiment is correct? It A. Does not have the factor to be tested. B. Has the factor to be tested. C. Is not necessary in scientific investigations. And D. Gives the investigator a lesser degree of confidence. Now, the main reason why we have a control experiment is to be able to make accurate judgment using a control experiment. So the factor to be tested need to be present in both the control experiment and then the actual experiment. The only difference is that the conditions for the actual experiment will be deprived of in the control experiment to use that as a measure. So if you are giving condition A, B, C to the main experiment, you don't give it to the control experiment in order for you to see whether the factor which you are being tested, or the factor you are testing, what will be the impact if the conditions are not available in the control as compared to the actual experiment. So our answer over here is it has the factor to be tested, so the answer is B. Now we have to look at the diagram that we've been given over here. This is a section of a leaf, cells of a leaf. We have to look at it and use it to solve question number 9. So the line XZ, as we can see over here in the diagram above, represent an actual distance of 10 centimeters and the length of the diagram is 5 centimeters. What is the magnification of the diagram? So we have A, okay, the magnification is times 0.2, B times 0.5, C times 2.0, and D times 50. Now if you look at the diagram, they're saying that the actual distance is 10 centimeters. The length of the diagram is 5 centimeters. To calculate the magnification, it is the length of the diagram on the actual distance. So the length of the diagram over here is 5 centimeters on the actual distance, which is 10 centimeters. So 5 divided by 10 will be given as a magnification of times 0.5. So the magnification over here is times 0.5. Question number 10. The body symmetry of an organism can be described as A, a section that runs through the length of the entire or part of the organism. B, a section that passes crosswise through the entire length or part of the organism. C, how a body can be divided into 2 or more equal parts. And D, when an organism is said to have been sectioned. Now the body symmetry, if we see the body symmetry of an organism, we are basically trying to refer to how a body can be divided into 2 or more equal parts. So the answer for question number 10 is C, how a body can be divided into 2 or more equal parts. That is what we call as the body symmetry of an organism. Now let's have a look at our last question for this tutorial. So the diagram below are illustrations, these actual pictures, of different sections of a particular fruit. Study them and answer question 11. So we have the orange over here with X, demarcation of X. Then we have another section over here with Y. Now question number 11, the sections in X and Y respectively are A, longitudinal and transverse, B, transverse and longitudinal, C, cross and transverse, and D, transverse and cross. Now if you've done section, you know that if you cut from the width or along the width, it is transverse, and if you cut from top to bottom along the length of the sample from top to bottom, equal parts, it is longitudinal. So our answer, the X is transverse and then the Y is longitudinal. So our answer over here is B, transverse and longitudinal. So thank you so much for watching, and I will see you in the next video. Bye bye.